Hello, good morning, everyone. I think we will start. This may join or not. Yeah. So in today's class, it's going to be a mixture of techniques that are left out. In the last class, I told you when we had a discussion on the through the syllabus, we identified some topics that were not yet discussed. So I will try to discuss. And I hope you all remember PCR, right? And here is a link. You can type this one and find out. There are many other types of PCRs that are there. Not that the overall principle of PCR will remain the same. Only thing, some of them we already finished. Something like, say, for example, variable number of tandem repeats we have finished, and AFLP, allele specific PCR, and many other things we have finished. Okay, there are other PCRs, other types. Mostly we also finished some of the important ones. The others remain. The other types of PCRs mentioned here are based on certain principle, but largely mostly about the objective of what they're doing. So they're not too different. You can go through it if you want to. I hope you remember what is inverse PCR. That is one thing we learned also. There are advanced techniques which we haven't discussed, something like digital PCR. You might want to study it. At this stage, we are not covering it in this syllabus. And yeah, we finished reverse transcriptase. We finished real-time PCR. Many other topics have been finished. But otherwise, please, you can go through if you want to, but it's not mandatory. They're highly specific for a particular purpose or so. And sometimes the same technique can be referred to by different groups in different ways. Sometimes that is also a problem. So because this is a topic in our syllabus, that is long range PCR. It is not a big deal, but we will I will describe the applications of it, and then we will go through a few things. Long range PCR, as it says, is about the size of the size of the amplicon. What is an amplicon? Amplicon is the region that we are intending to amplify, right? We then will have a primer design here and here, so that it will get amplified, and that size we are intending to amplify or the amplified part is called as the amplicon. So typically, say up to 2 kb or 3 kb, the amplification is good. 5 kb also can be done. But if you are intending to amplify very large sequences, maybe in the range of 10 kb and above, okay? If you want to have above 10 kb fragment, then there are going to be problems with the PCR. First thing is, we are going to typically use Taq polymerase. This is the cycle that is given. Nice picture to illustrate the PCR program. We have Taq polymerase. We have DNTPs. We have to add. We need to add buffer and MgCl2 and many other things. Of course, primers too. What would happen if you are to compare a PCR product amplicon size of, say, 1 kb and amplicon size of 20 kb? What will become the problems? Say, based on the number of cycles, primers is not a problem. Okay, primers is not that big a deal. It will be equal in both the scenarios. Are you seeing my sequence, my presentation, right? You're able to see the presentation, right? Good. So, and buffer is about influencing this. That's also not a big deal as long as you have the Taq polymerase. I mean, it's for function of the Taq polymerase. The number of DNTPs required in this case, I mean 1 kb amplicon and in a 20 kb amplicon. It might be sufficient in this case, but may not be insufficient in this case. I mean, normally also we usually have excess of DNTPs to allow the reaction towards polymerization. But then that could be one of the limiting factors. And I have to put in time required. Time required, assume we usually keep 1 kb, 1 minute per kb, right? So in this case, we will put 1 minute. You can't use 1 minute in this PCR. You have to use 20 minutes, right? It also depends on the type of DNA polymerase that we are using. For something like with high fidelity, that is one of the issues. If we are going to do that, the program is going to take longer, and the time and in the cycling may also influence the stability of the polymerase that we are using. Okay, and Taq polymerase enzyme required is usually fine for both the cases, but the amount of time or requirement in each round of PCR, there is a possibility for some of the enzyme to be degraded, and each 20 minutes you might have lesser and lesser Taq polymerase. So it is going to influence the enzyme is going to influence the in this reaction. So I will call this as reaction A and this larger amplicon one as reaction B. If we compare, we know what are the problems. The next other problem will be about mutations. If you can recall, Taq polymerase has a mutation, I think about 1 in some 10,000 or so, something like this. It does make mistakes. And if it is 1 kb, the probability of a mutation occurring, I mean misincorporation of a nucleotide, is very less, right? Mutation rate is very less. But if you are having 20 kb fragment you're trying to clone, then the mutation rate is very high, right? Because longer, the more probability of at least one misincorporation happening in each fragment is very high. So if we want to do long ones, we should be mindful of the time, extension time that we are giving, and about the stability of the polymerase, and also the fidelity of the polymerase. If you remember, we fall into a kind of dilemma. We don't want to have so much time. We want to reduce the time, but we want to increase the fidelity. If you recollect in one of the discussions where we have, speed is inversely proportional to fidelity, right? Typically, that is what in general philosophical terms do. That's what happens. The faster we tend to work, the more likely that we might make mistake, more probability of a mistake. So the same is applicable to majority of the DNA polymerases too. Taq polymerase has great speed, but then its fidelity is little bit compromised. If your amplicon size is small, then no problem, but otherwise it's big. If the amplicon size is big, then it is a problem. But if you take something like a high fidelity polymerase like PFU, they are very accurate, but then they are very slow. So what people do if they want to for long range issues. Before I move on to what I'm trying to say, I want you to recollect something like Paul 5 or Paul 4, right? I don't know if I discussed it or not. I think I must have. Translation DNA synthesis, right? I gave a phrase or a challenge. I asked you what is best. Nothing is better than something is better than nothing, or nothing is better than nonsense. In biology, what works is something is better than nothing, even if it is bad. And translation DNA synthesis, there is pole 4, pole 5, UMUC, and so on, UMUD. They do not have speed, they do not have accuracy either. No fidelity, no speed. But they are still useful for the cell. And that's the amazing part of that is one of the ways it demonstrates something is better than nothing. Okay. Now we want to, our objective is that we want to have a very large fragment amplified. Then we need to, the problem is we have to take care of the mutation rates and we have to take care of the time involved. So as a compromise, what people do is they use a mixture of Taq polymerase and something like one is a Taq polymerase, another one is a PFU, which is one of the polymerases with high fidelity. PFX and many other things are also there. In these kinds of long-range PCR, they tend to use a mixture of these two, so that this one probably reduces the number of mutations, but Taq polymerase tries to keep up the speed. Okay, but otherwise, long-range PCR is not. Sometimes you have to do. Then it is like a desperate attempt, but typically it's not. The products or the amplicons that are formed are usually not so useful for other purposes. You can probably use it as a kind of analytical technique, but not necessarily for cloning purpose. In the sense for sequence analysis and other things. If we are doing that, we have to take into account that there is a probability of mistakes in the sequences that we may have developed, okay, or amplified. That's about long-range PCR. Now, there should be another way of how I can clone. Say I'm talking about this is the large gene. If there is one gene and I want to have a clone because I need to produce a protein, that protein from it, how can I do that? Long-range PCR is helpful, but even if there is one mistake, there is something drastic difference change in the amino acid sequence there could be, and because of which there could be altered functions. I do not have a way of doing it. So one way is to do one of the ways at least is to do RACE PCR. It's the strategy is something like this. You can think of it that way. So here is we will call this as the 5' part and the 3' of the gene or open reading frame. Okay, something like this. So what the strategy is, if you can amplify this part, something like this, and amplify this one, say, using primer A and primer B, and if you can also amplify using primer C and primer D. So what we can get then is we will get two overlapping fragments, something like this, from one PCR reaction, from another PCR reaction using C and D, we can probably get something like this. And then we can mix it up. If you remember in one of the site-directed mutagenesis, in the two-step PCR site-directed mutagenesis, we get something like this. We mix anneal and then we try to generate a whole full-length gene. Okay. So in this, so each of these reactions, you can think of it as calling as RACE, rapid amplification of cDNA ends. In this in the strategy that I told, we are assuming that we know the sequences, and that is why we are able to generate a primer, right? If there is a gene, okay, there is a gene to which of which we only know a certain region of it. We know a small sequence of it. We don't know anything else about that particular gene. Then how will you do that? And for that, when you don't know and yet you want to generate these fragments, right? And then you can also end up generating the whole length also. So there are two things about RACE. First thing is, we don't know the whole sequence of the gene, and we want to clone the whole gene. Then how do we do it? Second thing is, if even if it is large or small, how do we approach this? So the given quantity, given information to you is only a sequence, a nucleotide sequence of a small region of a whole gene. We don't know where does it start. We don't know where does it stop. We cannot take DNA directly and do anything of it. Instead, we could actually take the mRNA. mRNA is also likely to have the same sequence, or at least the complementary sequence, and uracil in place of thymine. And why are we taking mRNA? Because mRNA transcription must have started somewhere. Open reading frame must have started somewhere around here, and there will be a stop codon. And that is why it can give rise to a protein, right? So how, I don't know where does the 5' start of this open reading frame. I don't know the 3' end also. So for that, what we can do is, I'll start with 3' end. That's the easiest one. That is 3' race. That means we are trying to figure out this region of the gene. So we isolate the total RNA. In the total RNA, we know the end of this one because it has poly A tail, right? So you can use oligo DT along with the linker of any known sequence, any known unique sequence you can give. Now we have using reverse transcriptase and the primer. This is the anchor primer. You're adding another linker region to it. So we have made cDNA of the whole, all the DNAs, all the mRNAs. Now we are going to have a sense primer. You don't have to call it sense primer for this protocol, they call it. Remember, I told you that there is a region of known sequence. Because we know the known sequence, we can use that particular region, a primer based on that particular region, and synthesize the second strand. Remember that this sequence is present only if it is gene specific, right? In the first one, we are generating the first cDNA strand of all the mRNAs that are present. Whereas in the second round of second strand synthesis, we are only doing it for our gene X or gene of interest because we are using specific primer. Now we know. Now what we can do is we denature, and now we can add the primer for the second one. I mean, we had this linker region, right, or anchor primer, whatever it is. We can add a primer that is specific to this, specific to or complementary to this region, and generate. Now we can use a primer that is the gene specific primer plus the anchor primer. And then the only product that will get amplified is the gene of interest, right? There could be, because of this region, there could be other mRNA or cDNAs polymerized too, but they will not be amplified at exponential stage. So when you run a gel, you might end up seeing one prominent band, and you may see some smears here and there, very light. So you can cut this out and do your processing or cloning or any of the purposes. Okay, I hope you understood that. The 5' race is a little bit difficult, or it is a little complex, because in the 3' race, we know at least one of the ends we know. It is going to be a poly A tail. But on the 5' race, we cannot figure out where it ends and what is the sequence also. So that's where it becomes a little bit tricky to do that. So here we start with a gene-specific primer. Of course, complementary strand or low strand you have to keep in mind. Gene-specific primer, and we have used reverse transcriptase and generated the first cDNA strand. Okay. Then we use terminal transferase. I hope you remember. Terminal transferase is a template-independent DNA polymerase, and its substrates are 3'OH and DNTPs. Whatever DNTPs you give, it will just add. So terminal transferase, when you add, here is a 3'OH, this is the 5'. So 3'OH, and you are providing you're adding terminal transferase and DATP. So ACE will be added here. Then you're going to denature. How would you denature or get rid of the RNA? Using alkaline conditions, right? RNA degrades itself because of the 2'OH in alkaline conditions due to formation of oxyanion at the 2'OH. Okay. Now we know, previously we didn't know what is the sequence on the 3' end. But now we have added terminal transferase and DATP, and there is a poly A added on the 3' side, and now I know the sequence of poly A. You can use G or any of those sequences. Now we can, because I know the sequence is poly A, I can use a primer with oligo DT and also have an anchor region so that you will now get the second strand synthesized. Remember this, the first cDNA strand is only made for the gene of interest, not for all, right? So we don't have the issues of having all the genes amplified. Some kind of issue like that is not there. So we can now use DNA polymerase, amplify it, and later use the primer again, gene-specific primer and an anchor primer, and one will now get a product like this. We can then mix these two products, okay? Product generated by 3' PCR and 5' PCR, and then perform PCR to get or to polymerize the ends so that you will get a whole region which can then be cloned and analyzed. I hope you understood that. 3' race and 5' race. Rapid amplification of complementary cDNA or complementary DNA ends, that is race, full form. Objective is, or the conditions or constraints is, it's a gene of which we know the sequence we know is only a small region. We don't know the 5' end, or we don't know the 3' end. In that case, how can we clone the gene? That's the objective about it, okay? Then there is another technique, it's about analysis of expression, and mostly it is about comparing two different sequences. I think I have it here. Okay, I will take it here and then I will repeat it in the next slide. So here the situation, differential displaying. That means when you say differential, it is about comparing two cell types or states or conditions. Say I have cell type A in normal medium, and just making up a situation, and cell type A in some minimal medium, say like that. And I want to know what kind of genes are expressed, what is the differential display or in terms of gene expression in cell A or bacteria A when grown in normal condition compared to that of when it is grown in minimal medium, right? There will be gene expression differences, and I want to find it. So the protocol is something like this. You will isolate the mRNA, and then you will add oligo DT, and you will get cDNA-like strands. And also you are going to add small hexa primers, hexameric primers. Sorry, it is something like random primers. You can have a mixture of hexameric parts. Those sequences or primers that you're adding can bind to any location. In the sense, if you say hexameric, the number of times the probability of it binding is very high compared to a normal primer, which is between 25 to 35 or 30 bases. Right here, the specificity is very high. When you say 6 bases, the specificity, in the sense, the probability of the complementary region occurring on mRNA is also high then, right? Or even on DNA. So, and also if you have different types of hexamers, then you are increasing the probability with which they can bind. So you have first generated the cDNA, first cDNA strand of all the mRNAs. Okay, you have those. And then now you are adding a random or hexameric primers like this, and they can bind anywhere in the different places. So each of them will generate a different sized fragment, isn't it? And when you take this pool and again you amplify using the hexameric primers and also the oligo DT, right? And then you perform PCR. Each of this reaction in each of these cases will generate a certain sizes of when you run on electrophoresis, you will see bands something like this. If the mRNA pool is different, has a difference, okay, that means, say for example, this mRNA is not produced and some other mRNA is produced, do you expect that there will be any difference between the two reactions, the first one that we did, assume this one, and the second one? If there is I hope you understand that the fragments displayed here are directly linked to the type of mRNA. Or what I meant is the nature of the mRNAs, qualitative differences. We are not going to, it's not possible, it's difficult to do quantitative differences, but qualitatively you can tell which one is present and which one is not present. So say in these two conditions, if you have this is a one present in sample, in this sample, but not present in the other sample. So now I can cut this out, and here is another that is present in this one. I can cut this out and try to, you can clone it or you can sequence it and then find out what is that sequence. And then you can figure out, if you know the sequence, and then you know which gene is expressed. So this particular gene is expressed only in the second sample, but not in the first sample, whereas this particular band is representative of a gene that is expressed in sample, in this sample, but not in this sample. Yes, that is differential display. It is by the starting material. The objective is to find out the transcriptional differences, qualitative transcriptional differences between two cell types or one cell type grown in different conditions or time points, developmental stages. It could be any of those. Okay, that's the objective of differential display. Here is another picture, which would probably, if you didn't understand in the other one, you could understand here. Yeah, basically the same thing. You have RNA, then you made cDNA, and you can have different types of things, and then you'll see that one of the bands that is extra will form a different set of bands. Then this can be taken out to identify which gene is expressed in that particular state and which is not. Okay. So the next thing I want to go is a situation like, 1 second, just be online. I want to have a slide here. So say You must have all, as a part of bioprocess engineering or so, you must have tried to isolate a protein or a compound. I have a cell and I want to isolate, I want a large quantities of a protein. Okay, I want to have it in very large quantities. In that case, how do we do that? Typically, because we are learning recombinant DNA technology, we would have made a recombinant DNA with in an expression vector with a strong promoter, and hopefully it will produce the protein. And that protein, then we collect all the cells, we will disrupt the cells and take all the proteins, right? A mixture of proteins, out of which one of it or some of it is our proportion. We identify in which we keep track of the protein of our interest based on activity or some kind of analysis using page and so on. And slowly we will go through gel filtration, ion exchange, and so many other things. And finally we get, we purify the protein. This is a lot of work, right? And sometimes it might not be possible for all kinds of procedures. Think of something like this. I have a cell. I constructed a recombinant DNA. And what if this protein is produced in large quantities and that protein is secreted out? How many things is it going to become, going to get reduced? All we need to do is, if this is the situation, we can grow in the bacterium in continuous cultures, not necessarily batch cultures like we would want it here. In this case, it would be batch. You grow, take out the cells, and then isolate and so on. I mean, purify the protein. So the bacteria are growing and they are secreting the protein outside into the medium. So I take the medium, I can centrifuge out the cells, take the medium, and now I have the medium along with the protein. And all I need to do is isolate or purify the protein, right? It would be much easier, much specific. Of course, the medium may also have other proteins, but it's not as many as the whole cellular proteins that are present. And that will make it much more easier for us to purify. So we need to just look at a few things before to understand. There are several secretion systems in bacterium. In bacteria, positive, gram-positive, and gram-negative as well. So there are type 1, type 2, type 3, type 4, and many other secretion systems are there, which will allow the protein to be secreted out into the system, into the medium outside, right? I'm not going into the molecular biology of all these secretion systems, but what we need to understand is that I hope you also remember how protein targeting is done in eukaryotes, right? Signal receptor hypothesis, or actually method or mechanism is there. So for the protein to be secreted out, sometimes it could be co-translational. Often it is not co-translational. In the previous ones that I've shown, the protein is already formed and then the protein is secreted out. You can also have it as a co-translational, where the ribosome binds here and secretes the nascent peptide, and peptide is secreted into you need a large complex of proteins, sec-A, sec-G, and so on, which will then secrete into the periplasm and from periplasm you will need other proteins to send it out, right? That means there is a way of how there is something about the protein or the ribosome to be recognized by the transport proteins, right? So it becomes simpler. I will take this, I will describe the same here. So you need some signal parts in the proteins that are secreted out. Not all proteins are secreted out, right? It's a selective secretion of proteins. For that reason, we need to know what are those signals. So in different bacteria or different secretion systems, the signals are different, and one can use those signals. So when you are cloning a gene, you can add the signal sequence here. So that will generate a protein with the signal system saying this is the signal. That signal is recognized by the transport proteins and will allow to be secreted outside the cell. Okay, but why do we need that is an important thing, right? It makes it very easy that we can generate a large, the downstream processing becomes much more easier because we have less things to purify. So I hope you understood this part, but again, it's not possible for all types of proteins. So for whichever proteins it's a possibility, this is a good strategy to do, and it will be really helpful in general. One of the things one needs to keep in mind is, say you have type 1, type 2, and type 3. Which of these is better? I would go with type 2. It is complex and all, but it makes it easier that the secretion signal is already removed here. That makes it much better, because that reduces one more step of removing the secretion signal. I want this protein X. I don't want to have this signal, a secretion signal attached to it. So between the secretion signal and the open reading frame of the gene, we would have put a enterokinase site or something, some sequence which can then be cleaved off by a protease, right? So if you have to choose type 1 or type 3, then we should have added a enterokinase site or some site which will cleave off, specifically will cleave off this part, the secretion signal. Then we will have the protein alone. But if you are choosing type 2, the secretion signal is deleted here itself. So the protein that is secreted in this case can directly be used for our purposes, right? It makes it much more easier. Yes. So the last topic is expression of lethal genes that I haven't discussed, I think. I will give you an objective. I want to have, okay, I will describe the situation, the difficulties, and probably from there you can try to solve it. There is a protein gene X. It produces a protein that is X, and if that is produced, the cell dies. And I want, say, if this cell expresses, say, 2 copies of protein X, then cell is killed. These are toxic proteins or lethal proteins. So we say lethal genes. Then my objective is to study the biochemistry of these lethal proteins. How do they function, so that we can probably use it for some purpose. Now my objective is I want to have 1,000 cells, 1,000 protein X. Okay, how can I get 1,000 X? The question is, say for example, I have 1 cell. That means, say, each cell can produce 2X, and then I will have 1,000. So now I have taken a beaker, culture medium, and put in 1 cell. If it is, this cell will become 2 cells. By the time, if the lethal gene is expressed, the cell is dead, and dead. The culture will not grow. So what should I do? In the recombinant DNA I'm making, okay, this is assume a vector. I have the gene for gene X, that is the lethal gene. And if I added, I'll need a promoter. It's an express, should be an expression vector, because I want the protein to be produced. If I add a constitutive promoter, that means X is always expressed, then what happens? The cell will die and the culture will not grow, right? So what should then my object, what should I do in the designing of this? I should have used a conditional promoter. That means I should be in control of the promoter, in the sense inducible and repressible. The more amount of, I should tell when it should induce and when it should not induce. So I have to use a inducible promoter. That is one thing when you are trying to express lethal genes. The second thing, then we will also have, we can also say this, now we have conditional, so we would have not added inducer. We would not have added inducer. So the cell will grow and divide further, and after many rounds it might become, say, 500 cells, and then we add inducer, right? And then in each of the cells, the gene expression will happen, and then you will have, if you purify the proteins, you'll reach your objective of 1,000 X proteins, right? That is one way of doing it. So it's all about how much control you have here. Some of them are toxic, even more toxic. Little bit of expression, and then they kill the cell, and then they get degraded. If I gave you a situation like that, now I'm saying X can be produced, right? Immediately after production, it will kill the cell and it gets degraded. Half-life is very less. How can I produce that? In that scenario, we need to make a translational fusion. I think I described that to you before about translational fusions. Say, what I should then do is to prevent the toxic activity of protein X, so that the cell grows and it produces more of X proteins. I mean, it is basically temporarily we are making gene or protein X, we are reducing its lethality. In that case, what we should do is we should make a translational fusion. So what will that produce? It will produce the, say, beta I added lac-Z. So it will produce lac-Z, a protein that is a combination of, or it's a fusion protein with both the subunits like this. This is protein X, and the other one is lac-Z part. Then we can produce as many as we want. There is no toxicity. Okay, so we can produce that. Then we can take out the cells from there, and if we are adding a translational fusion at the end, we don't want the lac-Z part in it, because it might interfere in the function of or the biochemical activity or interactions of X with any other targets or proteins and so on. So again, we need to put in a enterokinase site or anything that can be cleaved up at a later stage of downstream processing. We take this protein, and then we use the protease, which are specific protease we may have added to this, so that it will separate X, and lac-Z will be eliminated out. We can purify it using gel filtration or ion exchange based on our availabilities, and then now we can test its biochemical activities and so on. So there are two strategies of how you can make express lethal genes, and again, it is subject to the type of protein, the nature or the severity with which it functions. I mean, it's the sensitivity of the host cell to the lethal proteins. So one of it is about controlling the expression of it. The second thing is about trying to interfere with the toxicity of the lethal protein that is being produced. Okay. Yes. Do you have any questions? I will try to, and on Monday, I will take the rest of the class on the last lesson, I think. I guess I have finished almost every part. If there are any leftovers, please bring it to my notice. If there are also any topics that you want to be redone, that also you should try to bring to my notice so that we will have a quick discussion on them. Okay. Any questions?